Okay, good afternoon, everybody. So today I'm going to tell you about some of the work that I did while I was a postdoc at the University Medical Centre in Groningen in the Netherlands. And I became quite interested in transcription and translation and some of the bits in between while I was working there. So I don't work on a pathogen. I work on Bacillus subtilis, which is the gram-positive model organism. And it's become quite a paradigm for regulatory studies because it has these two very nice phenotypes of sporulation and competence. It has very large regulatory networks, including 16 sigma factors, 4 of which are involved in the process of sporulation, and 130 transcription factors, which can have a regulon of either one gene or several hundred genes. So today I'm going to talk to you about 3 different regulatory aspects that we picked out from a big omics study that I was involved in. And this includes the complex regulatory network surrounding a bacteriocin production in its immunity protein, the control of expression of an ABC multi-drug transporter, and also the use of a non-coding RNA to regulate a major transcription factor. So to express or not to express, that is the question. So this is an operon that encodes for a bacteriocin called sablancin. Sablancin is encoded for by this gene here called PsunA, and to get an active bacteriocin produced, you need to express all of these genes, which actually for the cell is quite an expensive thing for it to do. These are two quite big proteins, and they require energy for their function. PsunI encodes the immunity protein, and in order for this to be produced, this has to be made so that the cell doesn't actually commit suicide by producing this. So as I said, I was involved in a very large omics study where we looked at Bacillus subtilis being the transcription of Bacillus subtilis under 104 different conditions. So along the bottom of these graphs are the conditions. And the conditions include growth in standard media, sporulation, which is this region here, stresses, various different things. But anyway, there are 104 different conditions. And up the side, on the y-axis, you can see the level of expression of the gene at any particular condition. So what can we say from these two profiles? So this is the PsunA gene, and this is the PsunI gene. The PsunA gene is the expression is quite different across the 104 different conditions, which you could predict and say that a lot of transcription factors would probably be modulating the expression of this gene. And the PsunI gene, the expression, yes, okay, it changes a little bit, but it's more consistently transcribed compared to the PsunA, suggesting that it's not under the influence of so many transcription factors. And this is how I got into being interested in the expression of PsunA. What we did was we put the promoter of... Oh no. Okay, so what you should be seeing is growing bacteria on here, which we've put the promoter of sablancin under the control with GFP. And the cells would be grow. You would see no expression of GFP, and then all of a sudden GFP would be switched on, but it would only be switched on in a portion of the cells, showing that the phenotype of PsunA expression is heterogeneous. Okay, so this looks nice and complicated, and it is, and but all I want you to take notice of is this central region here, which is what is controlling the heterogeneous expression of sablancin. So we have SPO0A, which is the master regulator of sporulation and various other phenotypes Bacillus subtilis holds. And for SPO0A to become active and be able to repress or activate different genes, you require it to be phosphorylated. So once SPO0A has been phosphorylated, it is able to repress AbrB, which is the transitional regulator of Bacillus subtilis. So genes that are not required during exponential growth phase are repressed by this gene. AbrB also autoregulates itself by repressing itself, AbrB represses ROC, which is an H and S-like protein and binds to AT regions on the genome, so foreign regions of DNA. It also represses itself, and these two genes repress PsunA. And as you will see here, if you remove, so this is the wild type, and as you can see, this is quite heterogeneous because you've got very large bars. But if you remove AbrB or ROC from the genome, you reduce the amount of heterogeneity. And the heterogeneity is being caused by this autoregulation on their own promoters. If you remove AbrB and ROC from the genome, you reduce the heterogeneity of expression even more. So this is actually what you would have seen had I been able to show you the movie. This is time-lapse images taken from a growing microcolony expressing the PsunA GFP. So as you see, when it gets into stationary phase, some cells are expressing large amounts of the PsunA promoter, and other cells are not expressing so much of the PsunA promoter. And this is what you see for the PsunI. So the PsunA promoter is quite heavily evolved, but for the PsunI must be present in all cells, and it is homogeneously transcribed, so therefore all cells are protected from the expression of the sablancin. And therefore, because you've only got a few cells that are producing the sablancin, but you've got all cells that are immune, the whole population benefits from the expression of sablancin. And we think this is probably how, so I didn't say that the Sun operon is actually encoded on a prophage. And so the prophage remains in the genome because if it loses it and neighbouring cells are producing sablancin, they will die. So you would also predict that there will be no evolution of the PsunI promoter. And indeed, this is what we see. So if you look at the PsunA promoter, just to give you a comparison, it's not heterogeneous. It is heterogeneously expressed. And when you come down to the PsunI promoter, it's basically homogeneously expressed. And when we remove the same transcription factors as I described to you for the PsunA promoter, it's only when you remove ROC, and this is because it's binding to foreign regions of DNA, do you see any reduction in the heterogeneity or the homogeneity of the expression of this promoter. So therefore, all cells are protected. So now I'm going to go on to my next topic, which is sensing the need for expression. So as I've already described to you, protein expression production is very expensive for the cell, and it only wants to be making proteins that it actually needs at any one time. However, the bug also needs to be prepared for onslaughts from the environment, such as antibiotics or changes in pH or water availability. And so it needs to have regulatory mechanisms present that it enables it to do this. And it does this in a number of ways, including two-component regulators, anti-sigma sigma factors, and also riboswitches. Bacillus subtilis encodes 8 multi-drug transporters, and this includes bmrCD, which is quite heavily regulated, but we wanted to know how. So this is bmrC, which from the tiling array experiment that I showed you previously. And as you can see, it's not expressed, not expressed, not expressed, and then in these two regions you get quite high levels of expression. YheJ is a small open reading frame that is found upstream of bmrC, and you can see that, yes, it's transcribed at a higher rate than bmrC, and there's not so much variation in it compared to this. So we use the same principle as we used for the sablancin expression study, in that we put the bmrC promoter in front of GFP, and we looked and saw what happened when we added different antibiotics. The antibiotics that we found that this promoter was responsive to were those that targeted the ribosome, lincomycin being the one that activated it the most highly. So what you see here is we added the lincomycin at approximately 200 minutes, or equivalent to an OD of 0.5. And with the higher amounts of lincomycin, you end up with increased amounts of expression. But what you also need to notice is that we added it at 200 minutes, but the expression doesn't peak or isn't really switched on until 300 to 400 minutes, suggesting that it's growth phase dependent. And if we repeated this experiment by putting the lincomycin in right at the start, again, it isn't until 300, 400 minutes that we actually see maximal expression of this promoter. So if we look at the genomic context of bmrCD and then this little open reading frame, what we see is that there is a promoter upstream of YheJ, but there is actually no promoter upstream of bmrC. So what we'd cloned into our bmrC GFP promoter fusion was actually the promoter of YheJ and YheJ itself. So we made a second promoter fusion, just incorporating the promoter region of YheJ, and had a look again at the expression in response to different antibiotics. And as you can see, YheJ itself, the promoter, does not respond to antibiotics. So, but again, it is quite time-dependent expression. So the maximal expression is during stationary phase, which is similar to bmrC. The transcription of YheJ actually influences the expression of bmrC. So what we did was looked at the expression of YheJ, and once we saw increased amounts of GFP, we added lincomycin to our bmrCD promoter fusion. And as you can see, immediately, once we'd added the lincomycin at this time point, expression was switched on, suggesting that YheJ does influence bmrC. If we look in the structure of YheJ itself, what we see is that it's able to form a terminator, an anti-terminator, and an anti-anti-terminator. So under conditions where you are, when no lincomycin is present and the bmrCD operon is not actually required, the terminator and anti-terminator form. But when you add in lincomycin, the anti-terminator forms and allows the transcription to continue. So to test what is happening with YheJ, we made several constructs where we put the promoter of YheJ and we put the YheJ transcript under the control of a xylose-inducible promoter. And so the GFP is acting as if it's bmrC. So that acts as our readout. And what you can see is when we add xylose on its own to induce the expression of this, there is actually some read-through through this terminator, suggesting it's a little bit leaky. But then when we add the xylose and lincomycin together, we get a massive increase in the level of GFP, suggesting that our anti-terminator structure is forming and allowing transcription of the GFP to continue. And when we add lincomycin on our own, we don't see anything, which is what we would expect. However, when we truncate YheJ and remove the terminator region, we don't see this induction of the GFP. It stays at the same level as if it's just leaking through. So translation of YheJ is essential for the transcription of bmrCD at the right time. The next thing we made was a strain where we deleted the 5' UTR and the ribosomal binding site. And what we see is that there is very little transcription of the GFP when we do remove the ribosomal binding site. When we add it back in, we see that transcription increases, suggesting that YheJ is translated. And the final piece of evidence that we have to say that YheJ is translated is through using the erythromycin resistance marker on the plasmid where we've got the xylose-inducible promoter constructs. Now, erythromycin, when we add it into the medium, the gene on the plasmid modifies the ribosome itself and prevents or allows, basically blocks the action of these antibiotics and makes them resistant to it. So when we don't have the erythromycin present, we see this induction of the GFP, so the bmrCD is being transcribed and therefore translated. But when we add in the erythromycin, this phenotype is blocked. And what we think is happening is that the lincomycin is slowing down the ribosome. We've got transcription and translation that are coupled, and when the ribosome is slowed down, this allows the RNA polymerase to continue transcription of the bmrCD operon. So the final piece that I'm going to talk to you today is about the post-transcriptional regulation. Now, for a long time, it's been considered the central dogma of molecular biology that DNA makes RNA, makes protein. But this is no longer sufficient, in that many areas of the genome that were considered not to be functional do actually have function, in that they encode for non-coding RNAs and other such molecules. And this adds in a complex level of additional control to the bacterium. So in our tiling array study, we found about 1,500 non-coding RNAs, and of these, about 150 were small regulatory RNAs, and this includes S1022, which has quite diverse transcription across our 104 conditions, and this regulates AbrB, which also has quite a lot of variation in its transcription. So S1022 is this little open reading frame, or this little region here. It's approximately 130 nucleotides, and it corresponds to this region of the genome. So you can see that there is a promoter upshift, which is indicated by this little triangle, and the little square flag shows a terminator region. When we predict the secondary structure, we show that it seems to form quite a nice stable structure. And when we knocked it out, we spotted this. So the red is the S1022 deletion mutant, and the blue is the wild type. And when we grew it under different carbon sources, you can see that exponential phase is extended and that it has trouble getting into the stationary phase compared to the wild type. When we predicted potential targets for this, we came up with AbrB, and that it binds to around the ribosomal binding site and start codon of this gene. And this makes sense in that AbrB controls the transition into stationary phase. If we look at a Western blot of the levels of AbrB within the cell, if we concentrate on the blot grown in sucrose, as you can see, we've used BDBD as our consistently expressed protein, and AbrB is expressed to 100% in the wild type. If we delete S1022, we end up with more AbrB in the cell. We can complement it by adding back in the S1022 on the plasmid in the mutant. If we add in the same plasmid into the wild type, so we've now got 2 copies of S1022 in the cell, we reduce the level of AbrB within the cell. And if you remember back to the diagram where I was talking about the regulator of AbrB being SPO0A, if we delete SPO0A from the cell, we end up with about 100% more AbrB, and then we end up with 300% more AbrB, or 200% more AbrB when we delete the SPO0A and the S1022, which does suggest that S1022 is having quite a large influence on AbrB itself. The next thing we wanted to look at was to see the population dynamics of AbrB expression in an S1022 mutant. So we took advantage of a AbrB translational GFP fusion that we had in the lab. And if you look here, we have the black line showing wild type levels of AbrB. And when we delete S1022 from the strain, this shifts the level of GFP being detected to the right, which suggests that there is more AbrB being made, and it also changes the mean and the median of the expression. And then the wild type also changes the mean and the median of the expression and shifts it back to the left. So we can take advantage of this and calculate something called noise strength, where we take the median and square it, and then divide it by the mean. So as you can see on the left here, we have without S1022. As you can see, these spots are quite closely clustered to one another, suggesting that there is very little noise within the system. But when we add in, when we do have the S1022 present, this then expands and shifts up, suggesting that there is an increased amount of noise within the system. So an increased delta S1022 mutant increases AbrB levels and decreases translational noise. And increased levels of S1022 result in decreased levels of AbrB and more translational noise. So our little model of why this is happening is that this is the AbrB promoter region, and as you remember from the Psunl work, we have auto-repression of the AbrB promoter, and we also have SPO0A sitting on the promoter. So this region is quite complicated and quite full. Therefore, there is no other real way that you can control the expression of AbrB apart from by using this small regulatory RNA, which binds to the ribosomal binding site region of the AbrB transcript and stops it being translated. So S1022 increases noise in the system, and this ensures that the population is ready for all eventualities. So you want some cells that are growing very fast, so they're using up all the carbon source that is available, but you also want some cells growing slowly, which means that when the carbon source is used up, it's ready and able to fight all the things that the AbrB regulon involves, so things like proteases and sablancin. So science never solves a problem without creating 10 more. This was by George Bernard Shaw, and I think this is something that we can take away from our 104 condition. I think there's a lifetime's worth of work for I don't know how many people in that in those experiments. So whole cell transcription studies are very interesting, but will never tell you the whole story. And transcription does not guarantee translation, which is something that needs to be taken into account when doing these. So bacterial survival tactics is something that I'm quite interested in, and looking at single cells enhances our understanding of the system. And non-coding RNAs are a new level of regulation that require some in-depth study, and this is what I am going to be doing as a new investigator here at Warwick. So I have to thank the people that helped me with this, my 3 cake-eating boys, Shalka, Evad and Ruben. I also have to thank Pierre Nicolas, who led the 104 condition study and also helped us a lot with the non-coding RNA work. I worked for Jan Martin van Del when I was in the Netherlands, and the Basis Bio Consortium did the 104 condition work. And I thank you for your attention.
We're running